सॉरी मॅडम, आम्ही नवरात्रीच्या ग्रुप डान्सची आम्ही प्रॅक्टिस करत होतो.
Oh, so you were practicing for Navratra group dance competition. Very good, children. No need to say sorry. Taking part in competitions is a good thing. I have always stated that studies should go hand in hand with recreation too. And you know, children, the song which you have selected is all-time favorite with me also. Now, children, in this song, they are talking about हाथ की सफाई. A person who is going to manage 1, 2 का 4. And you know, in biology also, there is one magician और हाथ की सफाई करने वाला called as Meiosis. And today we are going to learn about meiosis. But before we start, we have to say a small sweet prayer.
God will make a way where there seems to be no way. He works in ways we cannot see. He will make a way for me. He will be my guide. Hold me closely to his side. With love and strength for each new day, he will make a way. He will make a way.
Students, right in the beginning of this episode, I have got one important announcement to make. See, all the episodes of Tension Not series are being shot in the classrooms which are not soundproof. That is why I advise you to use such earphones to get clarity of sound. Secondly, if possible, instead of watching Tension Not series on your mobile, watch it on your dad's laptop. So children, before our prayer, I started with magician in biology, and that is meiosis. ये जो बायोलॉजी का मैजिशियन है ना, वो एक सेल में से 2 नहीं 4 सेल बनाता है. That is why I call this process as the magician in biology. We have already studied mitosis, and we know that mitosis takes place in somatic cells and stem cells. Now these 2 types of cells have nothing to do with the process of reproduction. But in our body, there are some reproductive cells also. And these reproductive cells will not show mitosis, they will undergo meiosis. So meiosis takes place in reproductive cells. Now meiosis का शोध जो है, खोज जो है, वो खोज कैसी लगी होगी, या मैं जब सोचने लगती हूँ तब मुझे ध्यान में आता है कि In our human body cell, there are 46 chromosomes. 46 chromosomes are present in male parent also, male parent, and 46 chromosomes are present in female parents also. So when reproduction takes place, means there is union of male and female cells, then it should have been 46 into 2. But the zygote, that is first stage in baby's life, doesn't show 46 into 2 chromosomes. It shows again 46 chromosomes only. So there are 46 chromosomes in each parent and there are 46 chromosomes in child also. That means somewhere in between these two stages, the number of chromosomes must be reducing first. And that is why the child also shows 46 chromosomes. So 46 chromosomes in a somatic cell of a human being is maintained from one generation to next. That happens because of a wonderful process taking place in our reproductive cells, and this process is called as meiosis. So children, now we know that meiosis takes place in reproductive cells of our body. In case of plant world, the reproductive cells are called as spore mother cells. And in case of animal world, the reproductive mother cells are called as gamete mother cells. These spore mother cells or gamete mother cells, they show 2N condition, that is diploid nature. Now interface in these reproductive cells is just like the interface that takes place prior to mitosis. That means there is G1 phase, S phase, G2 phase, and in S phase, DNA replication takes place. So 46 chromosomes are there in mother cells. Now each chromosome is going to produce a chromatid, sister chromatid, and that is why we say that replication of DNA has taken place. So now the condition of number of chromatids is 46 into 2. Now, these 4 mother cells are going to take entry into meiosis, which is quite a long and complicated process. But for intelligent students like you, sky is the limit, and hence you are going to understand meiosis very easily. Now look at it. Meiosis is divided into 2 parts, meiosis 1 and meiosis 2. In meiosis 1 also, just like our mitosis, there is karyokinesis followed by cytokinesis. But since this karyokinesis and cytokinesis, they are taking place in meiosis 1, here they will be called as karyokinesis 1 and cytokinesis 1. Now, let us come on karyokinesis 1. Karyokinesis 1. The names of the phases are very easy. Why? Because they are like mitosis phases. Prophase 1, metaphase 1, anaphase 1 and telophase 1. What is not like the mitosis is prophase 1 is quite a long one and it is divided into 5 substages which we will be learning in our due course. Now during meiosis 1, in this karyokinesis, that is nuclear division, reduction is taking place. That means from one mother cell, 2 nuclei, each with N condition or haploid condition will be formed. This will be followed by cytokinesis 1 and hence 2 cells are formed. So up to this stage, that is end of meiosis 1, 1 का 2 हो गया. From one spore mother cell, 2 cells are formed, each with haploid condition. Now, we come on meiosis 2. Here again, it is subdivided into nuclear division, that is karyokinesis 2, followed by cytokinesis 2. And here the phases are termed as prophase 2, metaphase 2, anaphase 2 and telophase 2. Now, this meiosis 2 is more or less like mitosis. Why? Because it is an equating or equational division. The number of chromosomes was reduced to half, that is from 2n to n. But now, the number of chromosomes is going to be passed on without changing them. That means n number of chromosomes will be passed on. And here, from one spore mother cell, the end result is formation of 4 cells, each one with haploid or n number of chromosomes. Now, how this is happening? The entire process will be studied with the help of figures so that everything becomes easy for us to remember. So let's come in the next classroom to see more and more figures. Students, we already know that in meiosis 1, the phases carry the same name as in mitosis. That is prophase, metaphase, anaphase and telophase. But in meiosis 1, there are 5 substages. Those 5 substages I have shown on the blackboard. But before that, there is interphase occurring in mother cells, that is reproductive cells. And in interphase S, replication of DNA is going to take place, means chromosomes, each one is going to get 2 sister chromatids. Now meiosis 1, prophase 1, there are 5 substages, 1, 2, 3, 4 and 5. Below them, the scientific names given to these substages is written, but for S.S.C Boards, Maharashtra government state, these names are not included in syllabus. So you just have to write 1, 2, 3, 4 and 5 substages. Now what is happening in substage number 1? Chromatin network which was there before interphase, it gradually resolves. गुंता स्वतःचा स्वतः सुटलाय and the chromosomes are standing apart from one another. At this stage, with the help of good microscopes, you can actually count the number of chromosomes. And you will find that there is 2n or diploid condition, and the n chromosomes have come from our mother's side and another n chromosomes, they have come from our father's side. So the chromosomes which we have received from our mother I have shown with pink color, and the chromosomes which we have received from our father I have shown as blue color. Now here we can see that for every chromosome coming from father's side, there is another similar chromosome. See, for this chromosome, there is another similar chromosome coming from our mother's side. These two chromosomes are called as homologous chromosomes. Means they are similar in length, they are similar in their organization, and they are similar in the number and types of genes which they are carrying on them. So here is one chromosome from mother's side, here is homologous chromosome or similar chromosome given from our father's side. Now we come on this part, this we already know. These are two centrioles which are the cell organelles present in the cytoplasm. Now we come on the second substage. Here, homologous pairs of chromosomes come very close to one another and they lie parallel to each other. This condition is called as synapsis, and each pair is called as bivalent. So here I have shown one bivalent and another bivalent. Now let us see the substage 3. In this substage 3, we have shown that the chromosomes have condensed or shortened and their double nature is apparent now. Now these bivalents or 4 chromatids of homologous pair, they are present inside the nucleoplasm. And we know that nucleoplasm is a semi-fluid substance. So these chromatids of homologous chromosomes, they are going to flow like this inside nucleoplasm, and at some places they will come over one another in this fashion. That is what we have shown here. So where they are coming over each other, those points are called as chasmata. Now we come on the 4th substage. Here, a repulsive force for no apparent reason appears between homologous pairs of chromosomes. And up till now they were standing very close and parallel to one another, but now they want to go away, divorce लेना चाहते हैं from each other. So खींचातानी में क्या होता है? खींचातानी में some of the portions break at chasmata, and these broken portions are connected to another homologous chromosome. जो मदर से मिला, मम्मी से मिला जो भाग था वो आया है अपने पिताजी के क्रोमोसोम पर, एंड मम्मी का जो क्रोमोसोम था पिंक कलर का उस पे चिपक गया है पिताजी के क्रोमैटिड का एक भाग. So this is called as crossing over or recombination. And lastly, we come on substage 5 of prophase 1 of meiosis 1. Here, we will find that the nuclear membrane is gradually disappearing. The centrioles are coming up on opposite poles near nuclear membrane, and the chromosomes, homologous chromosomes with recombination part, they are still inside nucleoplasm. So this is the end of meiosis 1 prophase 1. Now let us see the figures carefully once again. Remember that half the chromosomes we have got from our mother, which are shown with pink color, and half the chromosomes we have received from our father, which are shown with blue color. After covering a big portion of meiosis, it is time to take rest in the form of musical break. Isn't it, Prashant?
येस मॅडम. मॅडम पण एक रिक्वेस्ट आहे, या म्युझिकल ब्रेकमध्ये आम्हाला थोडा डान्स ग्रुपची प्रॅक्टिस करायची होती, तर आम्ही करू शकतो का?
Oh yes, why not? Good idea. Instead of our regular musical break, you have to use this time for your group dance practice. Very good idea, Prashant.
थँक्यू मॅडम.
But Prashant,
येस मॅम.
there are two conditions from my side. Number one condition, keep the volume as low as possible so that other classes will not be disturbed. And secondly, allow me to watch your group dance practice because I am very much interested in music and dance programs.
ओके मॅडम, थँक्यू.
So let's have that break.
My name is Lakhan, my name is Lakhan, सजनों का सजन मेरा.
Very good children, very good performance. your enthusiasm, your energy was just amazing.
Thank you, madam.
Now, children, shall we start the remaining part of meiosis with the same enthusiasm?
Yes, madam.
Good. So come with me in the next classroom. Children, we have already seen the first 5 sub-stages in prophase 1. And this particular figure shows us the end of stage 5 in prophase 1. Now after this, in meiosis 1, there is going to be metaphase 1, anaphase 1, telophase 1, and they are somewhat similar to the corresponding stages in mitosis. Of course, there are some differences also. So let us see the similarities and differences now. In metaphase 1 of meiosis 1, just like in mitosis, the centrioles have occupied the opposite poles and they have sent spindle fibers, and the chromosomes are attached at their centromeres to spindle fibers in the equatorial region. All this is like mitosis, isn't it? But there is one major difference. In case of mitosis metaphase, the chromosomes were arranged on the equator in such a way that one chromatid of a chromosome was facing one centriole and another chromatid of the same chromosome was facing the another centriole. Now see what is happening here. Here, one member of homologous pair is facing one centriole and the other member of the homologous pair of chromosome is facing another centriole. Now let us come on anaphase 1 of meiosis 1. Here, just like mitosis anaphase, the spindle fibers are retracted. But you will find that centromeres are not split as it was the case in mitosis anaphase. Here, the entire chromosome is moving towards one pole and its homologous partner is moving to another pole. Now let us see what is happening in the telophase 1. Telophase 1 is like mitosis telophase in the sense that at each pole we are going to find the groups of chromosomes. But in telophase 1, every group of chromosome shows a chromosome with 2 sister chromatids. The chromosome which is coming to the pole is not a single thread-like structure, but it is double natured. Now the nuclear membrane appearance will form 2 nuclei inside one cell. After that, cytokinesis 1 will follow, which is exactly like cytokinesis in mitosis. So, these are the similarities and differences between mitosis and meiosis 1. Observe the figures carefully. see how after cytokinesis 1, two cells are formed at the end of meiosis 1. Now each of these two cells is entering meiosis 2 without interphase. No resting phase there. Now, meiosis 2 again shows karyokinesis 2 and cytokinesis 2. Now, karyokinesis 2 is very easy because it is just like mitosis. Only one difference is there. And what is that difference? Crossing over or recombination has already taken place and it will be passed on. Now at the end of telophase 2, we will find that in every cell two nuclei are formed. And this every cell will undergo cytokinesis 2 in the same fashion. So here at the end of meiosis 2 process, we will find 4 haploid gametes are formed. And these 4 haploid gametes, they will be participating in reproduction process. Students, right in the beginning of this episode, I said that in biology there is one magician called as Meiosis. हाथ की सफाई दिखा रहा है वो. क्या कर रहा है Meiosis? Meiosis में एक सेल से 2 सेल और 2 सेल से 4 सेल बनती है. How it is happening? That we have shown in a nutshell, that is in short form, on the blackboard with the help of this chart. Now this particular part represents the processes taking place in a male parent, and this particular part is showing the processes taking place in a female parent. Now let us first see what is happening in a male parent's body. In a male parent's body, the reproductive organs are testes. And in testes, there are reproductive cells. Each one of them shows 2n condition. Such cells first undergo interphase. So replication of chromosome takes place. Now please remember that in replication, one more chromatid is added. So first a chromosome was a single long thread. Now the chromosome has become double nature. That is, it has got one more chromatid after the replication of chromosome in S phase of interphase. All of us know that, right? Now, this particular cell enters meiosis 1. In meiosis 1, reduction is going to take place, and therefore the number of chromosomes will be halved. Half banenge number of chromosomes. N chromosome will be given to one cell, N chromosome will be given to another cell. Now, after meiosis 1, without showing interphase 2, meiosis 2 starts. And from one cell, two daughter cells are created. So this cell gives rise to these two cells. So ultimately, 4 cells are formed from one parent cell. Now these 4 cells are formed in the body of the male parent, and hence they are called as sperms or 4 male gametes. Now the same process is taking place in female parent's body. From one cell, first of all, in meiosis 1, 2 cells, haploid cells are formed, and in meiosis 2, 4 cells, which are again haploid, are formed. So 1, 2 का 4. ये हर एक पेरेंट में हो रहा है ड्यू टू meiosis. Now these male gametes, in case of a male parent, they are called as sperms, and the female gametes, in case of a female parent, are called as ova. In sexual reproduction, mating takes place and a male gamete unites with a female gamete. This process is called as fertilization, and the product of fertilization is a unicellular zygote, that is first stage in baby's life. So this end unites with this end, and 2M condition is re-established in the baby. So in the male parent, it is 2M condition. In female parent, it is 2M condition in all somatic cells. And even in baby, 2M condition is established right at the formation of zygote stage. Amazing, isn't it? And this goes on forever. So, 2M condition re-establishes after the fertilization process. Wonderful. Simply amazing. Students, we have covered meiosis. Now, we are going to cover 2 sure shot questions from meiosis. One is significance of meiosis, and the second is differences between mitosis and meiosis. The points I have already discussed during this episode. So now what remains is how to put the answer in our answer book. So I am going to give you ready-made answers for these 2 very important questions. Note them down. Students, now we have covered the question on meiosis. Now we will be seeing one very interesting 3D animation film on meiosis. And since we know the theory of meiosis, we will be enjoying this 3D animation film more. Watch out.
Join together at the centromere. They stay fused at the centromere throughout meiosis 1. Next, each pair of homologous chromosomes undergoes synapsis to form a complex involving 2 pairs of sister chromatids. Chromosomal material is exchanged between the 2 pairs of sister chromatids. This event is called recombination, or more commonly, crossing over. After crossing over, the sister chromatids for each chromosome are no longer identical to one another. This is one of the reasons why no two siblings, aside from twins, are genetically identical. There are several more key steps in prophase 1. The nuclear membrane begins to break down. Then, the two centrosomes migrate to opposite ends of the cell and microtubules appear. The microtubules then attach to the chromosomes. The next phase of meiosis 1 is called metaphase 1. Here, the synapsed chromosomes align at the equator of the cell. The chromosomes align randomly, which results in different combinations each time meiosis occurs. The next phase is anaphase 1. During this phase, homologous chromosomes separate and migrate to the two poles of the cell. Importantly, the sister chromatids remain attached at their centromeres. The final steps of meiosis 1 are telophase 1 and cytokinesis. Here, the cell divides into two daughter cells. Each of these two cells now undergo meiosis 2. Meiosis 2 is similar to mitosis. The first stage of meiosis 2 is prophase 2. Again, chromosomes condense, the nuclear envelope breaks down, and the spindle apparatus forms. The major difference between prophase 2 and prophase 1 is the fact that the daughter cells have only one copy of each homologous chromosome. So in prophase 2, there is no synapsis of homologous chromosomes or crossing over. In metaphase 2, the chromosomes align at the equator of the cell. Again, the alignment is random. Since the sister chromatids are no longer identical, there will be many different possible ways for these chromosomes to align. In anaphase 2, the sister chromatids are pulled apart as the microtubules shorten. Also, the ends of the cell are pushed farther apart as microtubules elongate. In telophase 2, the nuclear membrane reforms and the cytoplasm is divided into the two haploid daughter cells. This division is called cytokinesis. Since meiosis 2 began with 2 cells, and each of those cells were split into 2 cells, we now have 4 unique haploid cells at the end of meiosis.
So students, we have covered meiosis topic with significances and difference between mitosis and meiosis. Now, what is going to be our plan for the next episode? That is episode number 9. You know students that we are dealing with chapter 2 in your biology 2 paper. Now in the next episode, that is episode number 9, we are going to cover the remaining questions from chapter 2. For example, glycolysis, tricarboxylic acid cycle, and so on and so forth. Again, before we say bye, let me remind you that you are going to watch all the episodes of Tension Knot series with the help of earphones so that you get clarity of sound. Thank you very much. Take care of yourself and your family members.
हाँ चल के तुझे मैं लेके चलूं एक ऐसे गगन के तले जहाँ गम भी न हो आंसू भी न हो बस प्यार ही प्यार फले एक ऐसे गगन के तले